Hello friends, myself Syed Ibrahim Shah. Now we are going to discuss about the bacteria Yersinia pestis. Yersinia pestis is a biological weapon considered as one of the most serious bioterrorism threats. This gram-negative bacteria has been used as a source of aerosol weapon in the past. The bacteria also contributed in the World War as a biological weapon. Now let's get into the topic. General characteristics of Yersinia pestis. Grams classification. It's a gram-negative bacteria family Enterobacteriaceae. Shape rod-shaped bacilli. Motility non-motile in nature. Capsule and endospores are absent. Respiration facultative anaerobic respiration. In nature, optimum temperature 28 to 30 degrees Celsius and the optimal pH is 7.2 to 7.6. Habitat. Yersinia pestis has two main habitats in the stomach of various flea species at ambient temperature and the blood or tissue of a rodent host at body temperature. Alexandre Yersin, Swiss physician and bacteriologist, discovered the Yersinia pestis in 1894. Yersinia pestis is the hidden history of Black Death. Yersinia pestis is one of the most alarming pathogens throughout the century. Black Death was named after the clinical symptoms caused by the pulmonary plague. This infectious bacteria able to form a deathly plague causing high death occurrence. Now let's see pathogenicity of Yersinia pestis. Disease transmission. Yersinia pestis is an zoonotic bacteria usually found in small mammals. It is transmitted between animals through the fleas. Humans can be infected through the bite of an infected vector fleas. Incubation period 1 to 3 days. Virulence factors. First of all, we all should know what is virulence. The ability of the microorganisms to cause disease is called as virulence. The term virulence was first coined by the scientist Louis Pasteur. Virulence factor are the factor which are responsible for causing an infection to the human beings. The virulence factors of Yersinia pestis. Capsule, lipopolysaccharides, Yersinia outer proteins is also known as yolks. Plasminogen activators, pesticin and hemagglutinin proteins. Pathogenesis of Yersinia pestis. Yersinia pestis is a primarily a rodent pathogen. Humans are the accidental host by the bite of an infectious rat fly. Plague is the disease that affects humans and other mammals. It causes by the bacterium Yersinia pestis. Humans usually get plague after being bitten by an rodent flea that is carrying a plague bacterium or by handling an animal infected with the plague disease. While growing in the flea, Yersinia pestis loses its capsule layer. The most of the organisms are phagocytized and killed by the polymorphonuclear leukocytes in the human host. A few bacilli are taken up by the tissue macrophages. The macrophages are unable to kill the Yersinia pestis species and provide a protected environment for the organisms to synthesize their virulence factors. The organisms then kill the macrophage and they are releases into the extracellular environment where they resist phagocytosis by the polymorphs. The Yersinia pestis quickly spread to the lymph nodes which becomes hot, swollen, tender and hemorrhagic. The clinical diseases caused by this bacteria Yersinia pestis. Bubonic plague. Bubonic plague is the most common form of plague and typically manifests with the sudden onset symptoms. Bubonic plague is usually transmitted through the bite of an infected flea. The bacteria enters to the host bloodstream leading to lymph node infection. Fever, chills, headache, and muscle aches are the common systemic symptoms of bubonic plague. Then, septicemic plague. Septicemic plague occur when the bacteria spread to the bloodstream, leading to a rapidly progressing and severe systemic infection. Septicemic plague can develop from untreated bubonic plague or by the primary form of the infection. It can also result in the direct exposure into the tissue or fluid. Septicemic plague is life-threatening condition that can lead to the multiple organ failure and even death too. The laboratory diagnosis of Yersinia pestis. Microscopic examination. In gram staining, we can observe the pink colored gram negative rod shaped bacteria. Motility test, we can see that it's a non-motile in nature. Colony morphology on the culture medium. Blood agar. Yersinia pestis colonies are on the blood agar are typically non-hemolytic or gamma hemolysis. Then in McConkey agar, white colored non-lactose fermenting colonies are formed. We are using another selective medium for the Yersinia species. Yersinia cin agar. Colonies are on the cin agar often appear as pale to bright yellow colonies. The colonies may have a yellow center and with a clear edges. Biochemical test. Catalase test positive, oxidase test negative, urease test negative, indole test negative, methyl red test or the MR test negative, VP test negative, citrate utilization test negative, starch hydrolysis negative, casein hydrolysis negative. Nitrate reduction may be variable. Antibiotic therapy and prevention. Streptomycin after consider the first line treatment. It is effective against Yersinia pestis. Gentamicin is preferred as another option. Deoxycycline and ciprofloxacin are the alternative antibiotics for Yersinia pestis. Vaccination is the preventive measure for Yersinia pestis. Uses of insecticides, environmental sanitization, and measures to reduce the contact with rodents. These are all the preventive measures for Yersinia pestis. Thank you.